Kia ora. Good evening, everyone, and salam alaikum, and welcome to today's webinar, COVID-19 from virus to vaccination. Before we get started, I would like to go over a few items so you know how to participate in today's event. You will have the opportunity to submit text questions to today's presenter by typing your questions into the questions pane of the control pane, where there is Q&A box, but not the usual question chat box. You may send in your questions at any time during the presentation. It is expected that you send it only through the QA box. We will collect this and address them during the Q&A session at the end of today's presentation. So tonight, our guest speakers are Professor Vernon Ward, a virologist from the University of Otago, and vaccination experts Ms. Sally Schnauer and Ms. Amy Brass from Immunization Advisory Center. Before moving the presentation, I also would like briefly to acknowledge Pearl of Islands Foundation. Briefly, we call PIF. PIF is a non-profit charitable organization established by mainly New Zealanders of Turkish descent in 2006. PIF works comprehensively to celebrate diversity and advance social cohesion between cultures, to contribute to the educational, cultural, and overall social well-being of our community through its established institutions, platforms, and activities. So now I would like to introduce Professor Vernon Ward. Vernon Ward is a professor in the Department of Microbiology and Immunology at the University of Otago. He obtained his PhD in virology in 1987 and undertook postdoctoral research in virology at the Institute of Virology, Oxford University, United Kingdom, and the University of California, Davis, USA, before taking up an academic post at the University of Otago in 1993. Professor Ward has served as the head of department and subsequently as the dean of School of Biomedical Sciences at Otago. He has published over 100 referred research papers and book chapters in the field of virology, with his current focus being on RNA viruses, such as noroviruses. He currently leads an Endeavor Fund research program looking for new antiviral agents against a range of viruses, including SARS-CoV-2, as we know, COVID-19, that involves a multidisciplinary team from 7 research organizations across 3 countries. Please, Professor Ward.
Thank you. I'll go ahead and share my screen. And I'm hoping that's okay for everybody. Kia ora, everyone. As Sahiban said, my name is Vernon Ward. I'm a professor at the University of Otago in the Department of Microbiology and Immunology. So my brief for you today is to talk a little bit about the viral biology, and I hope that some of that will let you understand some of the things that are happening in and around this pandemic. So the virus we talk about is SARS-CoV-2, and we call that that name because of the symptoms that it's caused. So that's the virus, and the disease that the virus causes is called COVID-19. So the virus is SARS-CoV-2, the disease is COVID-19. And we call these coronaviruses because if you have a look at the virus under a microscope, then you can see you have this crown, or what's called a corona, of spike proteins sticking on the outside. So that's why these are called coronaviruses. Now, what I'm going to do with you today over the next 15 minutes or so is just touch bases on a few things around the biology of the virus and some of the things we know about the virus. Have a little look at where the virus might have come from. We'll have a little look at the disease that it causes, and then the virus itself and how it infects and the replication that it does. we'll use that information to try and talk a little bit about where these variants might be popping up from and how the replication of the virus informs the track and trace that's done with whole genomic sequencing. And then I'll finish up with a little bit about the tools we've got for controlling the virus and where this might go next for New Zealand. So that is the plan. So first question is, where do these coronaviruses come from? Or put it another way, how did SARS-CoV-2 get to humans? So you notice I'm talking about the virus, not the disease here. So in terms of where it's come from, the original source of this is most likely somewhere out of bats. Bats harbor a whole range of different viruses that are can be quite pathogenic to us and don't tend to do a lot for the bat. There's some metabolic reasons for that. And somehow the SARS-CoV-2 virus has got to us. Exactly how it's got there, we don't know. There's some various theories out there. The most likely route is that it's passed from a bat to some other animal and then to us. And that process of animal transmission is what we call zoonotic transmission. Once it's got to us, and it was probably somebody picked it up in a live animal market in Wuhan in China, although there is other theories out there. And once it's got to us, it turns out this is a virus that, while it's come out of animals, can transmit from person to person. And that transmission is reasonably efficient. And right now, the Delta virus, if you do nothing, no interventions, no masks, anything like that, then it's probably about one person will then infect 6 others. And then those 6 will infect 36, and that 36 will infect 216, and so on. And you can see how it spreads. The only other point I want to sort of make here is that we talk about viruses coming to us from animals, but of course, we can also pass the virus to animals, and a whole range of animals have been shown to have acquired this virus, and some of them have died from it, and some of them are just illustrated here. So SARS-CoV-2, it is dangerous, and just to illustrate that point, there's been over 260 million cases, and that's documented cases, so it's probably much larger than that. There's been over 5 million deaths, so to put that in context, that's more than the entire population of New Zealand. If you look at November alone, the last 4 weeks, there have been something like 200,000 deaths from this virus. So it is important, so it is dangerous. And the other thing is that it spreads very easily, and that's what I'm trying to show by using this image that came from a Science article last year. And what it shows us, what we know about this virus is that it spreads on droplets. So if you're sitting here speaking like I am now, or whatever, you're spraying droplets. Now, somebody could be very sick from the virus and they're coughing and they're sneezing, and that's effectively spraying the virus out into the environment where other people can breathe it in and get infected by it. The other thing about this virus, of course, is that a lot of people will catch the virus and not get any symptoms, but they're still very infectious. When they talk or sing, whatever they're doing, they are breathing that virus out into the atmosphere for others. And that's one of the reasons why we talk about using masks. And when you have a look at a mask, a lot of people think that a mask is about protecting you from catching the virus. Actually, the best benefit of a mask is to protect others. So think about your friends, think about your whānau. By putting a mask on, what you're actually doing is limiting the amount of virus you're putting out into the atmosphere. Now, it's never going to be 100%, but it can really minimize the chances of transmission and somebody catching it. So it does transmit easily, but there are things you can do to help with that. Having caught COVID, if you're unfortunate enough to have got the disease, then the virus gets to a lot of places in your body and causes a lot of issues. So you're probably well aware of the pulmonary issues and respiratory distress that people suffer, but it also gets in and affects your heart, it can affect your kidneys. It replicates as well in the gastrointestinal tract, and if you want to put this to what it means in a real world situation, because of this, it ends up in the wastewater stream, and that's why wastewater testing works, is because the virus is being excreted out into the wastewater system. Obviously, it can have effects, and part of the reason it can be very hard on people that have got diabetes is the hyperglycemia it can cause. And of course, it has a whole bunch of immune effects, inflammation, cytokine storm. So it's not very nice if you do actually catch it and are symptomatic. And it's easy to talk about, these obvious diseases and the fact that some people are dying, but actually there's also a lot more to it than just that acute disease. What you've also got happening is a thing that's called post-acute COVID-19 syndrome, or sometimes called long COVID. And we don't know exactly how many people are suffering from long COVID. The estimates I see range from about 10% to 30% of those that had an acute disease. So let's say 10% at the lower end of that estimate. people have long-term consequences from having caught the virus. So this is a diagram that's come out of this Nature Medicine article earlier this year, and it just shows what happens over the course of an infection. So somebody gets exposed to the virus down here, say it's week 1, it's a bit quicker for the Delta virus, and then the virus is replicating inside our cells, and you get more and more virus in your body, and it gets to a point where there's enough there that we can detect it with our detection systems. There's enough there that you now got an acute disease, but over time you get on top of the illness, and then of course you eventually lose the virus. And for many people, that's the resolution. However, for quite a few people, that 10%, 20%, whatever it may be, of people go into another phase where they have all sorts of problems. They struggle for breath and air, and they can't walk around the block. They get tired. They get this thing called brain fog where they can't think properly. They have headaches, and there's all sorts of systemic effects go on for these people. And this can go on in what they call an ongoing phase for weeks and weeks and weeks, and it's quite distressing for these people. They're past the COVID itself, but they've still got long-term problems. And then there's other people that go into a chronic phase, and it's showing here at 6 months, but there's people that have been suffering this for 18 months and more now. And these people that for a long time are called the long haulers. And we're still, I think, waiting to see just how big an impact it's going to have. And I guess the reason I've put this up is we think about the acute disease, we think about people dying, but there's a lot of debilitating effects linked to this infection as well. So, in order to cause all these effects, the virus has to be able to infect us. And one of the real critical things in virology is that a virus to replicate itself has to get inside one of our cells. It's not like a bacterium that can just be free growing or whatever. It has to actually enter our cells to replicate. In order to do that, the first step is the virus must physically attach to the cell it's going to infect. And that is one of the most critical steps. In this diagram image here, this flat surface at the bottom, that represents the surface of one of our cells. Sticking up from it is one of our proteins, and it's called the ACE2 protein, and that sits out on the surface of our cell. This big ball at the top, that's the virus. Now, this is a representation from real virus that people have imaged and then turned into this image. Sticking out from the surface of that virus is these club-shaped things. These are the spike proteins that stick out on the surface. And then highlighted here in this light blue color is a thing called the receptor binding domain. And that's the part of the protein that binds onto that ACE receptor and forms that physical attachment. Once that attachment has happened, it triggers a whole series of events that draws that particle into that host cell and it initiates infection. So that concept here from virology is that it must attach to a cell. In this case, it binds to ACE2. That's the absolute critical step in infection, and that's why vaccinating against the spike protein is such a good target, because if antibodies bind to this, then it can't bind to the receptor and it can't start an infection. Having bound to a receptor, the virus gets internalized. Our own systems draw that particle into the cell where it breaks down, and it releases the genetic material of the virus. And that genetic material of the virus is the viral RNA. And it serves two purposes. It makes viral proteins, so it's important. It codes for all the viral proteins, and those viral proteins get processed by a thing called a protease, a viral protease. And also those proteins replicate the genome, so they make many, many copies of the genome. And once enough of the viral genetic material has been copied, these assemble the proteins to make new viruses, and those new viruses hitchhike their way out of the cell and get released. And that virus is now free to infect more cells or to be breathed out and infect more people. So that's just an overview of that replication cycle. And just remember, the polymerase is really important for copying the virus, and the protease is really important for the structural components of the virus, the shell of the virus. So I want to touch a little bit more on that RNA polymerase, because it's actually really important for some of the things that you see in the big wide world, which is why I took a little bit of time over it. So this viral polymerase is what copies the genome. It's how the virus replicates. And what it does is it occasionally makes mistakes. Now, to try and illustrate that, the genome, if you will, of this virus is a whole bunch of nucleotide bases represented as A's, C's, G's, and U's. And there's around about 30,000 of these in one long strand in the genome. When the polymerase is making copies, every now and then it makes a mistake. And I'm just showing here where the A base has been changed to a U. So that's a genetic or mutational change in the coding sequence of the virus. So what happens is around about every million bases areas, what you end up getting is a little bit of an error. And if you think about it, each cell that gets infected by a single virus, if you look at this box at the bottom, produces about 1,000 viruses, and there's 30,000 bases per virus. So you're getting this tick over of this quiet genetic fingerprints of mutations happening in the genome because this is a polymerase when it's copying the viral genome that makes an occasional mistake. And of course, that gives you a pool of virus, and that's where these variants come from. You get these random, by dumb chance, really, variations, most of them no benefit whatsoever. And every now and then it'll be important. And what's happening, and this is what's happened with Delta, they end up being a change in the spike protein so that it bound the ACE2 receptor better. So it transmits better, it replicates a little bit better, it infects a little bit better. So it becomes a predominant strain. So that's where your variants are coming from. It's mistakes being made during viral replication. The good part of these mistakes is that it effectively fingerprints each virus, and you can tell during the track and trace and cluster determination where a virus has come from because it has a little bit of a unique sequence about it, and so you can track this virus. So there's some good sides to it too. So that's where the variants come from. That's how we know where somebody is likely to have acquired a virus from, or whether the two cases are linked or not. It's from the genetic makeup of the virus and the little mistakes it makes that fingerprints each specific strain. So having got to that point, I just want to take you back a little bit and then draw some of this together and what it means in terms of how we react to the disease. So first thing is that knowing that transmission route is really important to our public health response because it tells you what masks do and why masks are potentially important. Now, I'd like you to think over the next day how often you touch your face. You won't be aware of it until I've now said it, but just think about it, and that will tell you why one of the reasons why hygiene and washing your hands is really, really important. And of course, distancing. If this virus is being sprayed out into the atmosphere, if you're further away, it's harder for the virus to get to. If you're outside, then it's going to disperse a lot easier. But if you're inside a closed environment, because of this mechanism of transmission, it's more likely to transmit. And don't forget, it's about protecting others more than yourself. We now have vaccines, and whether it's whole virus vaccines, parts of the virus, genetic material, you can see why the spike has been targeted as one of the key elements for a vaccine response, because it can stop the virus before it ever gets inside our cell. What we're now starting to see happen is there's some very specific drugs starting to be developed to try and treat the virus, people of the virus once they've actually got it. Just recently, you've seen the announcement from Pfizer that they've got a new pill that you can take orally. Basically, it's pop a pill, quite a few pills in fact, and there's a protease inhibitor, that enzyme that I said is really important for making all the viral protein, and Paxlovid blocks that step of the virus replication. There's also new polymerase inhibitors coming online like molnupiravir, which stops the replication of the genome, again dampening down the virus. And of course, I want to give a shout out to our health professionals, because when you look at the actual disease, you don't only have to target the virus itself, you can target the actual disease. And that's where they've worked out that dexamethasone, for example, can dampen an inflammatory response and help with symptoms, giving people oxygen, get them to lie on their tummy. There's a whole raft of things that our medical professionals have worked out that can actually help ameliorate and help people get over the disease. So there's a lot of tools coming online that just weren't there 18 months ago. And this is my last slide before I pass over to my vaccination colleagues. And I guess it comes from the fact that I was asked to make a little comment to where I think it's going to go in New Zealand, but also because I got this question and I thought it's really important. It makes a great segue. Isn't this over? Almost over thanks to vaccination rates. And I've got both a bad news and a good news story here. So bad news. Bad news is no. We do have quite a way to go yet. So I've done some data, which I've pulled off a website here, of vaccination rates around some places in the world. And what I've presented here is the vaccination rates for the total population. So we often talk about the vaccination rates for the eligible population. Here in New Zealand, I'll just check this is still accurate, reasonably accurate today, is we've got around about 75% of the total population have seen at least one vaccine shot, and most have had two. And if you look, that's very similar to some of these countries I've highlighted with arrows here from Europe, we are in a very similar situation in terms of vaccination rates, but they're clearly having a lot of trouble right now with the coronavirus. I've also put up some of our nearby island colleagues and friends, and you can see they're on a path to get very similar vaccination rates as well. So we're all getting to very similar vaccination rates, which is great. But just remember that this will mean at this particular percentage of the total population, a quarter of New Zealand will not be vaccinated. So we will see a lot more cases. That is inevitable, and we're about to change to a new system and some more freedoms, particularly for our Auckland friends, and tomorrow, in fact. So, and we're going to see new variants. The good news, of course, is those tools that I showed you, and we're in a position now where the number of people that die are nothing like what was happening 18 months ago. The spread, people that have the vaccine have very good protection. It's not 100%, but the odds of ending up in a bad way if you're vaccinated are very slim compared to being unvaccinated. So, there's a good news story here. But don't think that we are out of the woods, and it is great that everybody's getting vaccinated, and the more that gets vaccinated, the better off we will be. So that's the good news story. There are things here that will help. And that's all I wanted to say. So thank you very much for listening, and I will now stop my share and pass to my colleagues.
Thank you very much, Professor Watt, for your very informative and enlightening presentation on coronavirus. It was a rich of useful information and clear information about the virus. Thank you very much once again. So now I would like to introduce Ms. Sally Schnauer to make her presentation on vaccination. Sally Schnauer is a New Zealand registered pharmacist working for the Immunization Advisory Center. Her role is to provide education and support to organizations that are promoting and administering COVID-19 vaccination. She has experience in health education sector and have a special interest in supporting vaccination while respecting the values and diversities of our people. Thank you, Ms. Sally, for joining us. Please. There you go.
Thank you very much. I hope everybody can see the screen there. Tēnā koutou no mai, haere mai. It's a pleasure to be invited to talk with you tonight about COVID vaccination in New Zealand. And I'm going to follow with some information about COVID vaccination following on from Professor Ward. So we're going to talk about what's in our vaccine, how well it works, and the efficacy and safety. For the most part, the Pfizer vaccine, which is our primary vaccine that we're using here in New Zealand. But I will also finish with a couple of points about the AstraZeneca vaccine, which has recently been introduced into our vaccination rollout. So let's get underway. Ooh, we're not going anywhere at the moment, for some reason. There we go. So to start with, what is in our Pfizer vaccine? The first is the antigen, the little mRNA. This is the active ingredient. It's the ingredient that sends a recipe to trigger our body's immune response to make antibodies. And antibodies are the fighters that kill the virus. They recognize that spike protein, as Professor Ward explained so beautifully. They recognize it should that virus enter your system and fights it off straight away. It also contains fats. These are actually little lipid bubbles that surround and protect the little mRNA. And to reassure you, the fat is not animal-based. It also contains sugar, and that's similar to the sugar that you pop in your tea or your coffee. And the sugar helps to keep the vaccine stable at the very cold storage temperatures. And it also stops the bubbles from sticking together and the lipid bubbles from sticking to the side of the glass vial. And the other thing that is in the vaccine are salts. They again help stabilize for storage, and they balance the acidity to make it in sync with the rest of our body. So also very important to talk about what is not in the Pfizer vaccine. It's not a live virus. So that means it can't possibly give you COVID-19 disease. It can't enter our cell nucleus. The cell nucleus is like the brain of the cell. It's a tiny little bubble inside the cell, and that's where the DNA lives. The Pfizer vaccine, with its little mRNA, can't penetrate the cell nucleus. It can get into our cells, but it can't get anywhere near the DNA. So for that reason, it can't actually alter your DNA in any way. There's also no DNA itself in the vaccine. There are no microchips, no human or animal cells, no blood products, no fetus material, no pork products, no gluten or soy, not any antibiotics, and in fact, not even any preservative. So in some ways, it's quite a plain vaccine. Many people ask us how the vaccine has been developed so quickly. It really is amazing to see what the world can do when all countries collaborate and are all on the same side wanting to get rid of this dangerous virus. COVID-19 was a real global problem, and many countries poured in millions of dollars into finding a vaccine to prevent this disease. So there was lots of international collaboration, and some of the steps were run in parallel, which means while they were investigating and designing the vaccine, they started building manufacturing plants, for example, which cost millions of dollars. But they were able to do that because the money was there. So they built manufacturing plants, made the glass vials, made sure there were plenty of syringes and needles, all the things that we need, not just to develop the vaccine, but also to administer it. So that really sped things along. But it's important to know that no steps were missed. We have an organisation in New Zealand called MedSafe, and that's our medicine safety authority. And they completed their own process, not just relying on overseas information. And MedSafe continues to review our New Zealand data. So all the information about people who have already been vaccinated here in New Zealand, they look at that and they are constantly reviewing that. So really important to know that we take the safety of the vaccine incredibly seriously. And the Pfizer vaccine has been used in 115 countries around the world. So a huge amount of vaccinations have taken place already. So how well does the vaccine work? And we need to talk about the Delta variant because that's what we have here right now. And it is the most dominant strain of the vaccine around the world. So we've got some information from the UK, and this information is quite useful for us because there are millions and millions more people in the UK than we have here in New Zealand. So they have seen using the Pfizer vaccine that it reduces the risk of infection by around about 80%. It also reduces the symptomatic disease. So that's the coughing and the sneezing, people shedding the virus and transmitting it to other people. So that's around 80 to 90%. But most important is the very high chance that you will not be hospitalized or die should you get COVID disease if you have been double vaccinated. So that data is really good from the UK, but you know, we need to talk about New Zealand. That's where we are. That's here, that's now, and this is current for us in our country. So this data is from mid-November, and it is talking about our eligible population. So it doesn't take into consideration our 11-year-olds and under, but for those people eligible to be vaccinated, that first bar shows that around 80% of us have been double vaccinated. It's crept up a little since mid-November. 10% of people shown there in orange have had a single dose, so they are in the system, and about 10% have not had any vaccination at all. So transferring that to our outbreak of cases, the blue area shows us that around about 67% of our cases are people who haven't had any vaccination at all. 20% have had one dose, but those that have had their full 2-dose vaccination, only about 12%. They're only representing about 12% of the current cases. And really importantly, those that are hospitalised, only 5% of the people in hospital have had a double dose of the vaccine. Around 20% have had one dose, and 3/4 of all those people in hospital have not had any vaccination at all. So important, I think, to relate data to us. So we like to fully inform people when they come in for their vaccination. And I'm a vaccinator. I work at the airport and vaccinate people. And I always inform them that there are possible reactions after being vaccinated. And the most common is to have some pain or redness and a little bit of swelling at the site where you had your injection. So that's usually the middle of the top part of your arm. Some people will feel a little bit tired, maybe have a headache and body aches for a few days. And these are things that we know can happen. So to a point, they're expected reactions to the vaccine, although not everybody feels that way. Some people are, as we say, right as rain. The reactions can be a good sign. They're not necessarily a bad thing. They can show that an immune response, your body is creating that immune response that we need it to do to fight off COVID should it come your way. So recently we've had a few changes in our vaccination rollout. We're always adapting and doing the things that we see are needed. And there are a group of people in New Zealand who are immunocompromised. What that means is that they don't have a strong immune system to fight off infection. And that can be for a variety of reasons. So we have introduced a 3rd dose of COVID-19 for very severely immunocompromised people 12 years and older. And those people will know who they are. They're usually people who've had, for example, an organ transplant or have had some cancers and are on some significant medication. And they will be talking individually to their doctor. These people are at a higher risk of becoming very unwell if they caught COVID-19. And they might not produce a really strong response from 2 doses. So we've introduced a 3rd dose. It's separate from the booster shot. If you can see in the little table down there, I'm fortunate that I'm not immune compromised. So I'm part of the general population and I have dose 1 and dose 2 with a minimum of 21 days apart. And then nothing for 6 further months until I get my booster. For someone who is deemed to be severely immune compromised, we give dose 1 and 2 in the same way. And then 8 weeks after dose 2, they will have their 3rd dose. And then 6 months after that, a booster. So 2 dose booster doses. So people since Monday, just this Monday gone, have had access to have a Pfizer booster dose. And I actually had mine on Monday this week, because I was one of the people who had my vaccinations quite early on in the year, being a health worker. So a booster dose sort of charges up the immune protection that has been provided by those first 2 doses. And it's available for anyone who completed their vaccination course, as I said, 6 months after their 2nd dose. And you can walk into a vaccination clinic, whether that is a pharmacy or your GP or a large centre, or you can use the Book My Vaccine website to make an appointment. It's really important for us to talk about pregnancy and breastfeeding. The Pfizer vaccine is recommended at any stage of pregnancy. Pregnancy or pregnant women and their babies are at much higher risk of serious COVID-19 hospitalisation, leading to intensive care and harm to their babies or their babies being delivered early, much more so than non-pregnant women. And in fact, data from, again, the UK tells us that 1 in 6 COVID patients receiving the highest form of life support are actually pregnant women. So we've had large scale surveillance data right the way around the world, and we've seen no increase in problems for pregnant women compared with non-pregnant women having the vaccine. So all the findings have been really reassuring. But pregnancy is a special time in people's lives, and so if anybody is concerned, we certainly endorse and encourage you to talk to your GP or midwife. The same with breastfeeding. We've supplied the Pfizer vaccine to pregnant women right throughout from the beginning of this rollout with no safety concerns, and the protection that the mum gains from being vaccinated passes through to the baby through breast milk. So the vaccine itself doesn't, it gets metabolized and it disappears from your system once it's done the job of making those antibodies, which is not much different from mum's antibodies from infections that she's had in the past going through to the baby to give them a good, safe start in life. So who can't have the Pfizer vaccine? We definitely don't give this vaccine to anyone who's had anaphylaxis. And anaphylaxis is a nasty allergic reaction. So anybody who's had anaphylaxis to an ingredient in the vaccine or their first dose of the vaccine, we would pause their vaccination. Currently also, anybody who's under 12 years old in New Zealand, and we're awaiting approval for children 5 to 11 years, but planning is underway. So that's great. We've been waiting to get robust data from overseas. We're fortunate here in New Zealand to be able to see what happens across the world in advance of us doing some of these things. And I think approval for 5 to 11-year-old children is quite close, and we should be able to deliver that vaccination early next year. And as I said, I would just touch on the AstraZeneca vaccine. The Pfizer vaccine does continue to be our primary or our main vaccine used in New Zealand, but AstraZeneca vaccine is an alternative option. And it will be for people who can't have the Pfizer vaccine for medical reasons, although there's not too many of those people, or people who need to be immunised because of the nature of their work and have any reason not to have the Pfizer vaccine. So it's basically giving people a choice. And the AstraZeneca vaccine is available through a limited number of sites now. A lot of people ask us about how to talk to people about vaccination. There's a lot of information out there. Some of it is good, some of it perhaps a little bit of misinformation, and it's really hard for people to work out where to find information and what information to trust. So not everything is always factual and accurate, especially some information that we can find online. And it's easy to accept information that aligns with our beliefs. And we can sometimes negatively react to news that is different. And just because an article might look good or read well, it doesn't always mean that the quality of the information is reliable. When people, I guess, express concerns about vaccination, we try in our roles to be really open to hearing people's point of view, to invite them to share their thoughts and show them that you will listen without judgment. We focus on the positives that we want to convey rather than just dismissing their concerns and beliefs. So we might, for example, talk about the care and love that we have for protecting our community, about why vaccination matters to me and why I care about that. Most people actually do want to protect vulnerable people in the community and support people close to them, their whānau, their friends, and their community in general. But I do think it's really important to remember you don't have to, you and I, we don't have to convince people. It's not our job to do that. And we do need to protect our existing relationships with friends and family. But those are just some tips about how to go about it if you're in a position where you can do that. We don't want it to lead to fights and arguments. So know when to call it a day. So just to finish off, we've got a wee slide there that does give you some information, some links to show you where you can go to find some good and helpful information. There's not a lot of time to read all of those out, but I know that this presentation is being recorded, so you'll have an opportunity to go back in and have a wee look at this and know where perhaps you can go to find little bits and pieces that is important to you. So thank you very much for listening. I hope that some of the information has been helpful for you, and I wish you all a safe journey as we open up here in Auckland and we get ready to enjoy a summer and a lovely Christmas. So I'll stop sharing my screen now, and I'm hoping that we're going to be able to have some time to help with some questions.
Thank you very much, Ms. Sally Schnauer, for your very educative presentation on vaccination. You made a big update in our vaccination knowledge. Thank you once again. Now it is time for a Q&A session. However, before starting the Q&A session, I would like to introduce another guest speaker, Ms. Amy Brass, who will support Q&A session today together with other speakers. But yeah, Amy Brass is a New Zealand registered nurse working with the Immunization Advisory Center in the COVID vaccine education program. She is passionate about education and working together with multidisciplinary teams to improve health outcomes in our community through vaccination and knowledge. She comes from a background in clinical education, emergency nursing, and primary care. Thank you, Amy, for joining us tonight. We are now going to begin answering the questions submitted during the presentation. As a reminder, you can still submit your questions through the Q&A box. So, questions, let us see which we have. One person, Trace Graham, asks about Novavax. Just maybe briefly, if we have any update about or any information about Novavax.
So I can make a start on that, and if either of my very esteemed colleagues would like to add to it, feel free. Novavax is a vaccine, another type of vaccine, and it's actually not being used in any country so far. So I know a lot of people and I'm not sure why, but I know a lot of people are kind of talking about Novavax or maybe potentially waiting for Novavax to come to New Zealand. But at this stage, it's not actually being used in any country. So it may come in the future, but I don't think it would be anytime soon. So those people that are waiting for Novavax, I think it would be worth considering looking at Pfizer and AstraZeneca as more viable options, because being that it's not being used in any country, we just don't have that real world data as well and that real confidence in it. Not to say it's not a good vaccine, but yeah.
Yeah, thank you very much. Another question is from anonymous person. So how does increasing vaccination rates affect the herd immunity? How does increasing vaccination rates affect herd immunity?
I can try and help with that one. So the more people that get vaccinated, and in getting vaccinated, you are less likely to have symptoms. So the more people who are vaccinated means that when you have a whole group of people and the virus is around, it can't take hold in like 90% of the people. So it kind of starts to die off. It doesn't have any success. It can't reach the unvaccinated in the same way. And different infections have different percentages of the required herd immunity. So some infections require like 90% of people to be vaccinated to have that effect, others maybe slightly less. May not be the best explanation, but I'm trying to keep it as clear as I can.
Yeah, I think I might just add a comment that part of what you're trying to achieve is limiting that spread, as Sally was saying. And the more people that are vaccinated, the harder it is. You know, all of these things, nothing is 100%. But if you think of that transmission rate, everything you do that knocks that transmission rate saves people getting infected. It makes, vaccination does make a huge difference.
There was actually a little study, it was actually not a little study, a study done in Sweden about household contacts and what happened when a family member got vaccinated. How did did they transmit to other people in the household who weren't vaccinated? And I think from memory, it was about 1.8 million people in the study over 80,000 homes. And if one person was vaccinated in the household, it reduced the transmission by around 40 to 50%. But the more people that got vaccinated in the household, the greater that, no transmission happened. It went right up to over 90%. So, if you had everybody in the household vaccinated and only one person unvaccinated, it reduced the transmission by over 90%, went as high as 97%. So that's kind of an example of herd immunity, but in a tiny space.
Yeah, and the other example of that is if you have 2 unvaccinated people in a room and 2 vaccinated people in a room, the chance of transmission is 200-fold lower where you're vaccinated.
Yeah, thank you very much for your comments. Another question is from Matthew Clark. The question is, for long haulers, is there a test they can take? As the PCR will only record a positive result during their infectious window. This may be important to workers with ongoing symptoms that don't have the proof of a positive PCR test.
That's probably my one, I think. That is an absolutely wonderful comment, absolutely on the button. That is exactly the problem for the people that are struggling with this. Not everybody's going to believe it because I still feel sick. There's a group being formed in New Zealand now, and there's groups being formed around the world to try and, recognize these people. So it's, there's a lot of them. It's very hard for them. You can't just take a PCR test, as you quite rightly pointed out, and say, yes, I've still got COVID. It's just there's a lot of them, and it's very hard for them to really prove it. some people have good employers that recognise it. Other, some people find it very hard. They can't work. As I was trying to say, I was being a bit cautious in just how big the problem is, because we really don't know. But it's going to be very hard on some people, and they'll struggle to work. They'll struggle to run a normal life. But most people do get over it over a period of time. But it's tough for them. So yeah, it's a real problem, and your question is absolutely on the button.
Yeah, thank you very much. Another question is from Jasmine Ituata. She says that it was hard enough getting vaccinations. Then there will be a booster shot. What is the likeliness that there are going to be more shots needed after this? The booster probably.
So, I'll start, and Vernon, you might have any more information on this. Look, we don't know is the true answer at the moment. We are in that fortunate position, as Sally talked about, that we are able to sit back and watch what's happened around the rest of the world a little bit. They all went first. They're all well into their booster campaign, but still not long enough now that we've seen waning antibodies enough to vaccinate again. The reason that we may need another booster in the future is if there's enough mutations in the virus to have to tweak the vaccine to ensure that we get better coverage, but we just don't know yet. So it is really important to, once you finish doing your 2 doses, to get that booster 6 months after your second dose. You did say, Jasmine, that it was hard enough getting your vaccination. Not sure what you mean by that, whether it was hard to access it. I know that maybe if you were in that June-July group, we did have a little shortage, but there is plenty of vaccinations now and plenty of places you can get it. And also plenty of places that will outreach. Lots of GPs are doing it now. But yes, we don't know at the moment what the future will look like in terms of boosters. Yeah.
Yeah, and if I just add a little bit, just from the virus perspective here, like obviously people have got their eye on this Omicron variant at present. A lot of the sensationalist headlines, we just don't know that we don't know what it's going to be yet. We just, this mutation thing I was talking about, people say, well, the virus is going to get less virulent over time. Well, we don't know that. It didn't happen for smallpox or measles or any of those sort of things. So why would it happen here? Delta is more virulent than the original. Of course, the next mutant that goes through might be quite benign. We don't know. And when people say very definitively what that outcome is, it's, really, we don't know yet. So will we need different vaccines? Omicron's got a lot of changes in the spike protein, right at the point where it interacts with the receptor. Well, it's got changes in points of the protein that the vaccines actually target. But what does that mean in terms of the need for another vaccine? We don't know yet. It might still be absolutely fine, but that's something that you just have to monitor, and we don't know yet.
And something to be reassuring is that the whole world, all of the experts in the world are looking at this. So, the information will come as fast as it can, as quickly as they can see the stuff, where it'll be shared. No one's sitting there hoarding all their secret information. And one thing that would be great to stop this virus mutating would be to improve some of the global inequities and be able to widely vaccinate our poorer nations, because when it is spread rapidly, it enables it to mutate faster than if it was a highly vaccinated population where it just couldn't get through those hosts. So hopefully we can spread the love and make sure that all our poorer countries are vaccinated.
Yeah, it's important.
Another question we have in usual chat box from Saba Majid. What positive and negative effects of vaccinations on pregnant women?
Okay, I can probably touch on that. There's been a huge amount of pregnant women who have had let's talk about the Pfizer vaccine, because that's what we have here. An enormous number of pregnant women who have had the Pfizer vaccine, and there have been no noticeable problems at all. So, in fact, no negative effects on having the vaccine when you're pregnant. What we have seen which is incredibly sad, is that pregnant women are very vulnerable if they get COVID-19 disease. It's led to a lot of preterm births, a lot of babies in intensive care, a lot of mums in intensive care on the highest, most severe life support. And it's led to loss of mums and loss of babies. So the midwives associations across the world, as well as obstetricians and gynecology specialists, are actively encouraging women who are pregnant, planning to be pregnant, to get vaccinated even more than a non-pregnant person.
Thank you very much once again. Another question I have here is, masks have been shown to cause hypoxia. So what comments could be done for that one? Hypoxia, the deprivation of adequate oxygen. So
Sorry, I can't see the question. Could you repeat it for me?
question is, this is my question actually.
Oh, okay.
It's not my own question from other members, so I have good. So yeah, I hope you can make some comments.
The comment was, the question was, masks are shown to cause hypoxia. What do we think of that?
Yeah.
Yeah, look, I think masks, it would be very rare for masks to affect your oxygen levels. Even people that have severe kind of obstructive respiratory disease would still adequately be able to wear a mask. If you felt like you were really, really hypoxic from it, you can actually go and see your GP and they can start the process of trying to get an exemption for you to wear a mask. But to be honest, most people, the majority of people that can kind of function in daily life would be fine wearing a mask.
Yeah. Thank you very much. Yeah. So I think now we came to the end of today's webinar when taking the time. So thank you, Professor Vernon Ward, Ms. Sally Schnauer, Ms. Amy Brass, and I thank you everyone for attending tonight's webinar, COVID-19 from virus to vaccination. If you have any other questions, please contact me. I will try to forward to our panelists. I hope they can answer our future questions. So you will receive a follow-up email within next 2 days with a link to view a recording of today's webinar. So on behalf of PIF, Pearl of Islands Foundation, and our presenters, thank you for joining us today and have a great evening. Thank you very much. Thank you.
Thank you.
Thank you. Have a good evening.